So today we are going to discuss about the CRISPR, which is a gene editing technology. In the gene editing technology, basically we are making some specific changes in the gene, or we can say we are doing some sort of editing in the gene. So gene editing cannot be done just by CRISPR. It can be done by Talon's technology, or it can be done by the zinc finger nucleases technology. So first of all, we will understand about these two technology before we move on to the CRISPR. For example, what happened in case of the ZFN, which is a zinc finger nucleases, they have basically two domains. One is the domain that binds to the DNA, and the another one is the domain that bind to the endonuclease. Okay, but this FOC1 endonuclease, until and unless they are not forming homodimers, then this ZFN cannot do any sort of editing in the gene. So the chances of the off-target effects are going to be less. And what are off targets? Target means, suppose if in your gene you have the sequence which is ACCG, and you want to do any sort of editing in this sequence, and the same sequence, let's say ACAG, is present somewhere else, there is only a one base pair difference. So there are chances that this ZFN can cut this DNA but can also cut this DNA sequence. So this is known as the off-target effects, and basically we all want to reduce the off-target effects because our purpose is just to cut this sequence, not this sequence. So this is known as the off-target effects. So here you can see, until and unless the FOC1 is not forming homodimers, this ZFN cannot make any sort of editing in the DNA to which you want to add it. So basically you need the left ZFP and you also need the right ZFP. Right ZFP will bind to your coding strand and the left ZFP will bind to your non-coding strand. Before we move forward, I want to clarify about the coding and the non-coding strand. So what is the coding strand? The strand that codes the strand with sequence is exactly similar to your RNA. And that is why it is also known as the non-template strand. Okay. Similarly, if we talk about the non-coding strand, the strand that work as a template. That is known as non-coding strand. For example, if suppose this is your sequence, if this is your sequence, A C T G G C A T C G, and then it will be T G A C C G T A G C. Again, this is going to be your 5' end, and this is going to be your 3 prime end, and similarly here it is going to be your 3 prime end, and here it is going to be your 5 prime. So can you tell me which one is going to be your coding strand and which one is going to be your non-coding strand? So this particular sequence is going to be your coding strand, and which is also known as the non-template strand. Okay, so don't confuse, and it is always denoted by positive sign. And this is your non-coding strand, and this is your, or you can say uh, template strand. Okay, so this is how, and why we are saying this is the non-coding strand, because RNA is going to be just complementary of this. So that is why it is the non-coding, but it is working as a template for the RNA polymerase to make the RNA, and that is why it is known as the template strand. Just to clear your basic. So you need to design two sequences. One is the left ZFP, which will bind to the non-coding strand, and the right ZFP, which will bind to your coding strand. Similarly, for the talons also, here are the amino acids which will bind to the left talon. Left talon will bind to the non-coding strand, and the right talon will bind to the coding strand. So talons are nothing but the amino acids. Basically, the conserved structure, they are basically consist of 31 to 35 amino acid. All the amino acids are conserved except 2 or 3 amino acids, as you can see here, 1 2 3 4 5 6 7 8 9, which are responsible to bind to the DNA sequence which you want to add it. And again, because FOC1, unless it is not forming the homodimers, there won't be any editing of the gene. So chances of the off-target effects are going to be less in this technology. So here one more thing is that this protein is guiding the endonuclease to make the specific changes in the DNA. So protein is guiding the endonuclease, while in case of the CRISPR, instead of protein, RNA is guiding the nucleases to cut the DNA. Before we move forward, we need to understand some basic things about the CRISPR. So there is one terminology which is known as the protospacer adjacent motif, and the sequences which is near to this PAM sequences is the protospacer. For example, we can say this is the sequence. This is the sequence which is the protospacer. Okay, and this PAM sequence need to be present in your template strand, or the strand or the DNA sequence which you want to add it. So this is the sequence need to be in your gene of interest. Okay. So this is the protospacer adjacent motif, and any sequence near to this is known as the protospacer, and this is a protospacer adjacent motif. Usually the protospacers are 20 to 22 amino acids long, and this protospacer adjacent motif, the sequence of the protospacer adjacent motif defines from which organism you have isolated your Cas9. Okay, so Cas9 here, this particular Cas9 is the endonuclease, and this is being isolated from many organisms. The most common organisms from which this protein is isolated is the Streptococcus pyogenes. And if in your experiment you are using the plasmid, in which the Cas9 is derived from Streptococcus pyogenes, it is necessary that, because this Cas9 will only recognize this particular sequence, which is NGG. Okay, so the Cas9 has two nuclease domain. One is the Reupsi domain. Reupsi domain that will cut the coding strand, and this HNS domain, this will cut the non-coding strand. Okay, and where it is going to cut? Just the 3 base pairs upstream. This Cas9 is going to cut the DNA sequence on the basis of these nucleaceous domain. For example, here you can see this is the structure of Cas9. This REC1, this will REC1 and REC2, they will recognize the guide RNA, and this one HNS and Reupsi, these are the nuclease domain, and this is the PAM interacting domain. Okay, so these are the basically, this Cas9 is a single unit enzyme and consists of many subunits. Here you can understand. So the Reupsi domain of the Cas9 will cut the coding strand, and the HNS domain will cut the non-coding strand. So there are some terminology which you need to understand. One is the PAM. PAM sequence is going to be in your gene of interest, but not in your guide RNA. This is your guide RNA. So whenever you are designing your experiment and making the gRNA, usually the softwares are being used, for example, the ChopChop or some other softwares. But if you are designing it manually, or if there is a question in the exams, then you need to understand that your gRNA will not contain any PAM sequence. Your gene of interest, if suppose this is the sequence you want to add it, this sequence need to be present near to the NGG. But again, this is just true for the Cas9. It is not true for all other Cas enzymes, which we will discuss in the future videos. But in case of the Cas9, and specifically the Cas9 which you are isolating from the Streptococcus pyogenes, it is necessary that it should have a NGG sequence in your gene of interest. Okay, and again you need to understand, one is the Reupsi domain, one is the HNS domain. Reupsi domain will cut the coding strand, and the HNS domain will cut the non-coding strand. Coding strand is basically the positive strand, and the non-coding strand are negative strand. So this is some basic terminology. And the another thing which you can see here is this one, the tracer RNA. And what is tracer RNA? So this is the tracer RNA. You need not to worry anything about it. You need to just design your gRNA that will be having the lower stem, and this lower stem and the tracer RNA will be there in the plasmid, which you need to purchase from any vendor. So tracer RNA usually, again, they are isolated from bacterial species only, for example pyogens or some other species. So they have the anti-repeats. What are anti-repeats? That is a partial repeat. So they are partially complementary to the lower stem of CRISPR-RNA. And this sequence is going to be conservative, means whenever you will purchase the plasmid, the sequence is going to be there. So these are the anti-repeat. This is the bulge region, and this is the termination region, terminator domain. So tracer RNA, if you are isolating from one bacterial species to two different bacterial species, and you are doing the modification or the mutation in the bulge region, you may see that in case of the one species, whole editing efficiency is going to be lost, while in case of the other species it is not. So it is very tightly coevolution. Okay, tightly coevolution. In one species, if you do the mutation in the bulge region, then there is no change in the editing efficiency. But if you are getting the tracer RNA from some other species and doing the same mutation, there may be the complete loss of the editing efficiency. So there is a very tight co-evolution, tight evolution you can say. Okay, so this is the basically tracer RNA. It is also very important. For example, you can see this is the tracer RNA and this is your guide RNA. Okay, and this is your gene of interest. Okay, so once this Cas9 will cut the DNA, so RUC domain will cut the DNA here and the HNS will cut the DNA here. So you will be getting the double stranded blunt end. Okay, so this point you need to understand. I just want to clarify you some basic terminology of Cas9. Now on the basis of this Cas9, CRISPR is being divided into multiple types. Before that, just understand what is CRISPR. So CRISPR, the discovery of CRISPR happened once scientists found that the bacteria is showing the immunity, which is the adaptive immunity. For example, if we are having some sort of infection, we are having the memory cells, and if the next time the same infection is going to happen, our memory cells will kill that bacteria or kill that infection. Similarly, what did the bacteria? They chop the viral DNA. When the virus is attacking the bacteria, they chop the viral DNA and integrate that viral DNA in between these repeats. These are the palindromic repeats. So what are the palindromic repeats? For example, if you are taking A, N, A, N and N, A, N. So if it is 5 prime to 3 prime or 3 prime to 5 prime, they are similar. So this is known as the palindromic sequences. Okay. So what bacteria did? Suppose this is the viral DNA. Bacteria has some palindromic repeat. They integrate that viral DNA in between these palindromic repeats, and then they make the, suppose this is DNA, genetic material of the virus's DNA. Then the bacteria make the RNA, which is known as the pre-CRISPR RNA, that will convert into the CRISPR RNA. And now it will bind to the Cas9. So here this CRISPR RNA, which is also known as the guide RNA, this is also known as the guide RNA. So it is guiding Cas9 to cut the DNA. Okay. So this is palindromic repeats. These are the spacer molecules, which is nothing but the viral genetic material, and then there are the Cas genes. So one gene is CSN1. Then the Cas1 or Cas2, and then the Cas9. Whole of these structure make the CRISPR locus. Okay. Now, on the basis of Cas9, from they are making up of one subunit or multiple subunits. CRISPR is basically divided into the mainly two types. One is the type 1 CRISPR, and another one is the type 2 CRISPR. So in type 1 CRISPR, what happened? The viral DNA genetic material is integrated in between the palindromic repeat, forming the pre-CRISPR RNA, and pre-CRISPR RNA is being cleaved to form the CRISPR RNA. So here you can see the palindromic repeats are forming the hair loop pin-like structure, and this is the viral DNA. Now it is combined with the Cas proteins. Here the Cas proteins are made up of the multiple units. Okay, so if suppose you have a plasmid, okay, if suppose you have a plasmid, so and it is made up of the 10 subunits, so you all the units should be there, 1 2 3 4. So your plasmid should have that much of packaging efficiency. So this is the drawback of this tri-bun CRISPR. That is why it's not commonly used in the laboratory. Okay, now this is the RNA and virus genetic material is, if the same, this virus is again attacking the same bacteria, then this viral DNA will combine with the RNA, which is complementary to each other, and Cas9 will cut the viral DNA. So here you can see the PAM, which is a protospacer adjacent motif, is going to be present in the viral DNA. Now coming to the type 2, the difference is like this, that in this the Cas9 enzyme, Cas9, whatever the enzymes are being used, for example Cas12 or Cas13 are also the type of type 2 CRISPR. The basic difference is that these enzymes are made up of the single unit, as you can see here. Single unit may be having a multiple subunit, but a single unit. Okay, and the another difference is the tracer RNA. Tracer RNA, as I already have discussed with you, but it needs not to be present in every type 2 CRISPR Cas proteins. For example, in case of the Cas12 or Cas13, it is not present. So this is just the basic of CRISPR that I discussed, and also I just want to clarify you. If you modify the RUPC domain, there will be no nicking in your coding strand. If you modify the HNS domain, there will be no nicking in your non-coding strand. So there are two types of mutation. For example, D10A, T is the aspartate converted into the alanine. Similarly, H is the histidine converted into the alanine at 840 position of the Cas9 proteins. If there is a mutation in the RUPC domain, there will be no cutting in your coding strand, which will be having the NGG sequence. Okay, and there will be cutting in the lower strand. So you will be getting something like this overhang. And similarly, in case of the, if you are modifying the RUPC domain, there will be no cutting in your non-coding strand. So you will be getting the overhangs. For example, here you can see there, this is the 5' overhang and this is the 3' overhang. If you have any doubt how to recognize which overhang you have, so just remember the longer strand, whatever the longer strand has at its left side position or the right side position, you can decide it. For example, in this case, the longer strand is this one and it has the 5' overhang. Here the longer strand is this one and it has the 3' overhang. So this is known as, and that's how you can recognize the overhangs. Okay, and also, as I said, so if there is a mutation in the RUPC domain, no cutting here, but there is a cutting here, so there is a single strand overhangs you will be getting. Okay, so this is also known as the nickage, and it is basically very good if you want to reduce the off target effects, and also the whenever there is a repairing, suppose if there is a double stranded break are there, then there will be the insertion and deletion of certain bases. But if there is a overhangs, then there is not much randomly addition and deletion of the bases, because this will work as a template for the other strand. So you can reduce the off target effects. You can also reduce the randomness of the indels, which is the insertion and the deletions, as you can see here. So there is a double stranded break. This is the non-homology and joining, where there will be the insertion and deletion. But if there is a single strand break, then the other strand will work as a template. So not much randomly addition and deletion will be going to happen. And this is the homology dependent repair, where you are adding the homology sequence which is complementary to this particular sequence, let's say here, and whatever the sequence you want to have, suppose if here this is TCG and you want to make it TCA, so you just modify this sequence. So here it will be, you will be getting, instead of TCG you will get TCA. So this is the homology dependent recombination, because you are adding you are adding the donor sequence, and which is homologous to your template string. So that is why it is known as the homology dependent recombination, and this is the non-homology end joint, because in this we are not providing any donor sequence. So with this I am going to complete this, and we will discuss about some more advanced CRISPR technologies in my next lecture. So thank you very much for joining me. Have a good day.